Now we will talk about the mechanism of siRNA action. When siRNA enters into the cell, how it performs its function. When we take the siRNA, it is mostly made available as double-stranded long molecule of RNA, 500 base pairs, or in the form of a hairpin loop molecule, double-stranded molecule that has a hairpin loop at its one end. Then this particular molecule is going to be processed by a set of enzymes which are found within the cell. Dicer is the enzyme that comes in action in the very first step, and it binds to the long double-stranded RNA molecule, cuts it down into smaller molecules of required length. And these smaller molecules are basically the functional part of RNA silencing or siRNA procedure. Dicer itself works like an RNase enzyme, RNase 3 enzyme, a class of enzymes which can cleave the RNA molecules. And an additional property that these enzymes target the restriction sites. They behave just like restriction endonucleases, and they target the restricted sites of specific sequences. Once the enzyme dicer has performed its function, siRNA molecule is made available of the shorter length 20 to 24 nucleotides or base pairs. Then comes another multi-protein complex which is known as RISC or RNAi induced silencing complex. This RISC molecule binds with the double-stranded siRNA molecule, and it holds the antisense strand of this siRNA, whereas the second strand, which is the sense strand of this RNA molecule, is separated and removed. Now, RISC is associated with the single-stranded portion of the siRNA. RISC looks for the complementary nucleotide sequences on the mRNA, which are present in the cytoplasm. Wherever the complementary sequence of nucleotide is found, RISC let the siRNA molecule base pairs with that messenger RNA, as can be seen at this step. After this base pairing is developed, then the endonuclease activity found with the RISC cleaves the RNA molecule from the space or from the required or specific sequence space. Ultimately, the RNA molecule is degraded. This degradation is going to cause gene silencing, and at the same time another option can be accessed here, that when this RNA molecule itself gets cleaved, it can be used as RNA interfering molecules again. इस हवाले से अभी हम एक एनिमेशन की तरफ मूव करेंगे. Now this animation will show you how the gene is going to transcribe RNA. RNA is being processed in the nucleus. Then it is going to be released into the cytoplasm. Where comes the dicer? From where comes the RISC? How the siRNA molecules are generated. The RISC is going to separate the two strands of the RNA molecules, the siRNA molecules, then looking forward for complementary sequences on messenger RNA and performing its function of degradation. After the degradation of messenger RNA, how those parts of the degraded messenger RNA can be used by the RISC to perform further subsequent gene silencing.